let me just tell you that what I've learned is if you let a baby play with your laptop, sometimes they will push a button that you didn't know was there. And that button will disable your web camera. So I've now learned that. So a little more careful about what happens to the laptop. All right, so we're going to start out as usual with our news from Mars, but there's not a lot today. This initial picture shows the helicopter in its new position where it landed after it did the one-way flight. And it hasn't flown again, and we don't know exactly when it will fly again. They'll be based on where the rover, whenever the rover feels like they need the helicopter to go off and get them some images of either things they might want to look at or hazards they want to not run into, then they will send the helicopter up again. I did want to point out one thing about this. As I said, this was at the end of its one-way trip, where it left its familiar area and came a couple of hundred feet at least and landed here. You'll notice that it's landed in, pretty good spot considering what's around it. Like there's these rocks over here, those rocks back there. And once again, that's just a tribute to the navigation software on that helicopter and how it can look at the images it takes of the ground and figure out where it needs to go. I want to say a couple more things about the helicopter. We have talked a little bit about the landing gear, mainly pointing out when we looked at the images taken from the helicopter that it shows the little feet that it lands on. But I wanted to say a little bit more about that. I think way back when we first talked about this mission, we pointed out that if the helicopter ever lands and falls over, then that's the end of it for the helicopter almost certainly, because the rotors would almost certainly get damaged and it wouldn't be able to fly again. So it's very important that it be able to land stably. Then there are two factors in that. One is the design of that landing gear. It's made out of carbon steel, which is very strong and very lightweight. And it has a built-in suspension system. The other thing that's important is the actual technique that they use in landing the helicopter. Now, you might think, since you're so concerned that it not tip over when it lands, that what you want to do is get over your landing site and then hover and slowly descend down until you touch the surface. But there's a problem with that. And the problem is the wind on the surface of Mars. We saw in a video last week a dust devil moving across the screen at a pretty good clip. So we know that the wind is capable of blowing pretty fast. They've actually got the wind gauge on the environmental sensor, and they know that the wind can go up to over 100 miles an hour. And so if you were trying to slowly land and you got caught in the wind gust, then the helicopter could be blown sideways and might like run into a pile of rocks like those that we saw. And so their actual technique is to come down at a pretty decent speed so that they don't risk hovering in the wind and getting blown away. And so they're depending then on that suspension system so that even though they may hit the ground fairly hard, they will absorb the shock and the helicopter won't tip over. So that's really, again, it's just a remarkable testament to the research that they put into developing this helicopter. Now, The other thing I wanted to do with the mission today is just show you some pictures. We mentioned that the rover has been examining some rocks, and what this is it's actually, that's not really a video, it's an animation. You're seeing the same thing over and over. This is the camera on the robotic arm, and what they're doing is they're zooming it in on this rock, and you see it getting more and more detail until finally, like right there, you're really like it's like you stuck your eye right up to the surface of the rock. So that just shows you the capabilities of that camera, and because it can do that, then they can decide, hey, this is, you know, this rock looks like we really want to find out more about it. And I picked one other picture, and that's this one. And the reason I picked this picture, I'm not a geologist, but I'm pretty well willing to bet you that what we're looking at here is a stream bed. Not probably it's a seasonal stream bed that back when Mars had a lot of water, that in the rainy season, water would rush down this way. And as it did, it would pick up these pebbles and deposit them along the way. If you ever do much hiking, especially after a heavy rain, it's easy to find something that looks pretty similar to this here on Earth. So once again, it's just an indication that at some point, millions, maybe billions of years ago, Mars used to have a lot of water and a lot of climatic conditions that we would recognize, similar to our own. Okay, so as I said, we don't really have any big news about the mission today, but I did want to mention that. And before we go on to the next story, anyone have any questions? Okay, then let's proceed along. Next story is also astronomy related, but it's not about Mars, it's about Saturn. And it says that Saturn's core structure has been determined. This story really interests me for a couple of reasons. We talked when we had our solar system class about the fact that one of the questions about the gas giant planets, Jupiter, Saturn, Uranus, and Neptune, is what exactly is their internal structure like? We know that they're made up of a lot of gas. In the case of Jupiter and Saturn, it's a lot of hydrogen, a lot of helium, and then some other gases. What we don't know is whether the planets have a solid core or not. Or rather, I should say, up till now, there basically have been two schools of thought about the structure of those planets. One says that the way the planets, the gas giants formed, is just that as the sun formed by the collapse of a big cloud of hydrogen, then there was leftover gas that was outside the part that collapsed into the sun, and it collapsed on its own. In the case of for the outer planets, and made these big gas balls that had the possibilities to become stars themselves, but there wasn't enough gas there. The resulting gravity of those planets was not enough to start the process of nuclear fusion, and so they ended up just being basically huge balls of gas. So that's one theory, and that says that there's no solid center at all to those planets, that they're just a gigantic ball of gas. The other theory says that there is a solid center, and it's the idea behind that, it's a little different picture of how the solar system evolved. It basically says that after the sun formed, that there was a lot of debris left in orbit around the sun that was mainly rocky material, and that over time the chunks of rocks would collide with each other, and some of them would stick together. And as they did, their gravity increased, and they would attract other chunks. And so you build a whole series of rocky planets. And this accounts then for the inner planets, for Mercury and Venus and the Earth and Mars. And then they said, well, okay, when we get out to Jupiter, something different happened, that we started with a rocky core, just like something similar to the Earth. But the difference is that Jupiter is 5 times as far away from the sun as the Earth is. And so the temperatures there are much cooler than they are in the orbit of the Earth or Mars. And so, whereas the Earth, for example, early on would have had a fair amount, picked up a fair amount of leftover hydrogen and helium gas, but that gas wouldn't stay, because it would be heated by the sun, and that would give it enough energy to overcome the Earth's gravity, and it would escape off into space. But the gas out at the orbit of Jupiter, it's much cooler there, so it has less energy, and so it could not escape from the gravity of Jupiter, so it stayed there. And in fact, Jupiter accumulated more and more gas, and so did Saturn. And that way you built up a big ball of gas around a solid core. So those are 2 different models that astronomers have come up with for describing those gas giant planets. Well, it turns out, based on some data, for Saturn at least, we now have a different idea. And this is interesting for 2 reasons. Number one, it's a different idea. It's different than what we looked at before. And number 2 is where the data came from. This is not new data. This is not something that's been collected in the last year or 2. You remember that NASA had a mission called Cassini, which orbited Saturn until 2017. It went into orbit in 2004 and stayed there until 2017, and did all sorts of measurements on Saturn and on its moons and on its rings, collected enormous amounts of data. Well, that data is still being analyzed because they had so much data. It's taken them a while to go through the process of analyzing it. And now some astronomers have looked at 2 different measurements that the Cassini orbiter took of Saturn. One was they measured the gravitational field of Saturn very carefully. Like every orbit they went on, they were continuously measuring what the gravitational effect of Saturn was. And so they have a very good map, for a 3-dimensional map of the strength of Saturn's gravitational field. They know how it varies. The other thing they did, well, they did a lot of things, but one of the other things they did is to take images of the rings of Saturn. And they noticed in particular that the innermost ring of Saturn, which is called the C ring, the rings of Saturn, they all have letter names, and they start with it goes like ABC, except that the C ring is the one that's closest to Saturn. The A and B ring, they got named in the order of discovery, and the A and B rings were discovered before the C ring, but the C ring turns out to be the one that's closest to Saturn. So in the data they took on the C ring, mainly just a bunch of images, they noticed that there is a wave in that ring, that the material in the ring actually oscillates up and down. And what they've done is they have taken the data on that wave motion of the rings and compared it to the data they have on Saturn's gravitational field. And they have reached the conclusion that the only way that they can account for those waves in the rings is that the material, Saturn's internal composition must be totally different than anyone thought before. It's not all gas. It does have a lot of solid material, but the solid material is not all concentrated in the core of the planet, down to lowest level. It's kind of spread out within the planet. It's true that the majority of the solid material is in the core, but there is a solid material that they can show that there's got to be solid rock, hunks of rock going out to occupy a total of 60% of the volume of the planet. Now, that doesn't mean it's all solid, but you do have these hunks of rock there that come out quite far from the center. And so they're now working on coming up with a theory of exactly how the planet could have evolved like this, that somehow it managed to accumulate rocky material, but for it not to all clump together. And they think, again, it's got something to do with the temperature out there, the fact that there was a lot, in addition to the rocks, that there was a lot of frozen gas out there. Water, for sure, we've known for a long time that there's water in Saturn's interior, and some other chemical compounds as well, ammonia, methane, so on. At the location of Saturn, the temperature is cold enough that those things will actually freeze. And so somehow, as Saturn was built up, this managed to build this kind of I don't know how to describe it. It's kind of like a Rocky Road ice cream. You've got a lot of frozen stuff, and then you've got these chunks of rock sort of embedded throughout. So this is really it's a brand new theory, and as such, it's very interesting. It's something that most astronomers had just not thought that you could have something like this. So it really is a step forward in our understanding of the solar system. Now, we don't know whether the same thing would be true of the other gas giant planets or not. And we probably can't find them by the same technique, because even though the other gas giants also have rings, Jupiter has rings, Uranus has rings, and Neptune has rings. But their rings, the thing that distinguishes the rings of Saturn is they're mostly ice, and they scatter light very well, which means you can really tell what's going on with them. Whereas the rings of the other planets are mainly dust, and it's much harder to get an image that will tell you, oh yeah, there's a wave going on here. So we don't know if we'll be able to, if the same theory does apply to the other planets, and there doesn't seem to be nearly as great a chance of being able to show, as was shown here, that it does, if in fact it does. One other thing that they think they've resolved here is a peculiarity of Saturn among all the planets in the solar system. Every other planet in the solar system absorbs more energy from the sun than it radiates out, or an equal amount at best. Saturn is different. Saturn actually radiates out more energy than it receives from the sun. And that's been another real mystery to astronomers. Nobody's understood why. And they think now, the guys who made this discovery think they may have the answer for that as well. because they say that when Saturn was formed, that there was naturally from all the collisions of hunks of rock and frozen water, frozen methane, so on. There's a lot of heat generated. That stuff collides with each other. Those chunks collide with each other, and that they think this particular distribution of rocky matter, the fact that rocky matter extends pretty far up into Saturn's interior, would cause it to retain a lot of that heat of formation. And therefore, it would still be able to radiate more heat into space than it receives from the sun. Now, if that's correct, then that sort of says that Saturn is unique in having a composition like this, because none of the other planets had this property. They all either take in more heat or then they radiate, or at best, they radiate back all the heat they get from the sun. But this is something that's definitely happens just with Saturn in the solar system. Okay, anybody have a question about this story? Okay, then let's go on. We're going to change gears now and go back and talk some more about DNA. We're probably in the future, and we've had at least one or two stories already regarding DNA. We're certainly going to see more of those in the future, because, of course, it's a very, very hot research topic in biology. Lots of people work on DNA. And so our headline here says, scientists show how transposons alter DNA. Well, what's a transposon? Let's first recall a little bit about what we learned about DNA when we talked about it back in our class on DNA and evolution. And one of the things we learned, and I don't know if I made the point so much then, but I want to make it today, is that everybody thinks, okay, DNA, we really got going learning about DNA when Crick and Watson made their discovery that DNA has a structure of a double helix. And that's a very important discovery. There's no question about that. But something I think is turning out to be equally important was actually discovered before the double helix structure was discovered. And it was discovered by an American biologist, a woman named Barbara McClintock. And we did talk about her earlier as well. She worked all her life at a biological laboratory on Long Island called Cold Spring Harbor. And what she did, all her research, her entire career, was studying the DNA structure of corn. And she was just fascinated by corn. And we actually talked a little bit about that. One of the reasons that she was so fascinated about corn is that she was able, with some other people, able to figure out exactly how corn has evolved. We know that it started out as a weed growing in Mexico, something called teosinte. And you can, I didn't get an image of teosinte today. We did look at one back when we talked about this. And it just looks like a weed, and it has a little structure at the tip of the weed that resembles a miniature ear of corn. And somehow over time, evolution transformed that into the plant that we know today. And Barbara McClintock, in her study of the evolution of corn, realized something. because she could look at the structure of this weed teosinte, and she could look at the structure of some intermediate plant's DNA, and then she could look at the structure of a corn plant, of the DNA of a corn plant that we have today. And what she realized was this, that certain genes that you could find at a certain location in the DNA of teosinte, when you got to modern corn, they were still there, but they weren't in the same place. They had moved within the DNA structure. And she thought this was truly fascinating. And in the, I think roughly around 1950, she published a paper in which she announced, hey, I have been able to see that somehow in the evolution of DNA, some of its genes have changed their location within the DNA structure. And when she did this, all the other evolutionary biologists who had been reading her work up to then thought it was a crazy idea, because everyone had the idea that DNA was a fixed structure, and they just, they thought she must be wrong in her claim that these genes had somehow moved from one location to the other. And she finally decided, that she wouldn't try to deal with, do more research on this and publish more about it, because people just weren't listening. They weren't looking at what she thought was solid evidence that her statements were correct. And so she just continued to do research, but just sort of let this particular topic alone. And about 20 years later, some other biologists who were studying bacteria discovered, lo and behold, in certain types of bacteria, exactly the same thing happens, that certain genes can migrate from one location to the other within the DNA of the bacteria. And once they published their work, then people had to say, well, wait a minute, we were all wrong and she was right. And thank goodness, at least, she did eventually win the Nobel Prize for the work she had done, because people recognized that she was the first person to show that this was true, that there are certain genes that can really migrate from one position to the other within DNA. At the time, those genes were called transposition genes. But now, because they're studied much more frequently, and transposition genes seems like too long a thing to say, people have started calling them transposons. So that's where the term in the title of our story comes from. Now, sometime after her discovery, Francis Crick, who had been one of the discoverers of the double helix structure in DNA, managed to show definitively that the purpose of genes was to provide a code for expressing proteins in cells. Remember when we talked about DNA, we said that DNA, that double helix structure, has 4 different amino acids. And just for, I won't bother with the names now, but the letters A, C, T, and G are the first letters of the names of those acids. And your DNA has just constant repetitions of one or another of those acids. And Crick showed that if you took 3 consecutive amino acids, that they would provide you a code that would tell you what kind of protein should be, when a cell divides and it gets a copy of the DNA, that DNA would read off the code, the sequence of A, C, T's, and G's, and tell the cell, hey, here's the proteins you need to make in order to function properly. So that's sort of that's the kind of a real quick summary of the basics of how DNA carries the information about the structure of cells. But when people started looking into it, they found out there's a little more to it than we first thought. First of all, when cells duplicate, the new cell does indeed get a copy of the DNA from the old cell. But it also gets something else. It gets some RNA, which is a sort of a cousin of DNA. It's got the same kind of structure as DNA, except instead of being a double helix, it's a single helix. But it has the same code pattern that the DNA does. And that it's actually the RNA that is really involved in producing proteins in the new cell. And the way it happens is when the new cell is created, then an RNA segment, which is called a messenger RNA, is produced in the cell. And then enzymes within the cell come in and they slice up that RNA into little strings of amino acids for proteins. But when they do that, it turns out that not all the RNA gets used. In fact, most of it doesn't get used for that purpose. There are sequences within the RNA that are given the name of introns, and they don't represent code for any kind of protein. And when they were first discovered, biologists thought that they weren't, there was no purpose, they were just there. But finally, after people studied the way the RNA functioned long enough, they came to the realization that these introns do serve a function, that they don't determine proteins. What they do instead is they're like little switches that have the property of turning genes on or off, so they can determine whether genes do their job or not. And so just like we changed the name of transposition genes to transposons, then they started calling introns transcription factors, because they sort of work to determine how genes are transcribed when a new cell is created by splitting off an older one. Well, that gets us a lot more along the road of explaining how DNA works, but it leaves a big question, and that is, how in the world did this very complicated process develop through evolution? Natural selection helps us understand some things about genes, that if we have a mutation in a gene and it now produces a different protein, and that protein turns out to be something that enhances the survival of whatever kind of life we're talking about, then chances are that that mutation is going to continue. If, on the other hand, the mutation produces a protein that either doesn't do any positive good or actually does harm to the organism, then that mutation is going to die out, because it doesn't help make this new version of the organism become successful. But that sort of accounts for the basic functioning of genes, but it doesn't tell us how these transcription factors got into the act. That just seems it's an enormously complicated process, and it's just hard to understand how evolution could have led to such things happening. Well, now we have a biologist at Cornell University, and they have suggested a way that transcription factors came into existence, and it is in fact that they're related to these transposons. It's been shown by this biologist and some others that when you have transposons, when you have genes that are able to migrate through the DNA structure, that occasionally they will merge with other genes and effectively create a new type of gene. And when they do that, then that means that the new gene that they create has a strong relationship to the original transposon, because it went into making up the new gene. And that makes it likely that the transposon could, in fact, perform an action on this new merged gene. So the theory here then is that these transcription factors were derived from transposons. And the key evidence for this, this kind of merged gene has been studied. It's been found, for example, that in the human genome, there are about 44 such merged genes. And if you look at other organisms, you'll find a fair number of them. And their test for their idea was to work with a gene in bats that they determined was a merged gene. And what they did was this. We talked about CRISPR a couple of weeks ago. It's a gene editing tool that can allow you to either remove a gene from an organism or install a new gene into an organism. So they took these bats and they used CRISPR to remove a merged gene. And they observed that the transcription factors that the bats had didn't work anymore, that they were no longer capable of turning on or turning off genes. Then they used CRISPR again, and they put that merged gene back into the bat's genome, and immediately the transcription factors began to work again. So that seems like fairly substantial evidence to show that these transcription factors are somehow related to the merged genes. And we know the merged genes are related back to transposons. And so that's why they think that transposons are the evolutionary predecessor for transcription factors. Okay, anyone have a question about that? Okay, let's get on to our next story. Back to astronomy. And this story says, title is A New Milky Way Supernova Candidate. And that interested me. Supernovas always interest me. And you remember back about a year and a half ago, I guess it was, there was a lot of talk about the possibility that the star Betelgeuse in the constellation Orion, we know it's going to one of these days be a supernova, but there was a lot of talk because of the way it was behaving that it might actually be something that was going to happen like soon, where to astronomers soon means, next 1,000 years or so. And it turned out that after a lot of research by astronomers, they're able to show, no, that's probably not true. That the changes that were observed in Betelgeuse, they think, are as a result of the fact that it has undergone a process that all big stars go through, in which it stopped burning hydrogen in its core and started burning helium. And so they think that's an indication that process has just sort of gotten going. And that would mean, probably it would be able to burn helium for another 100,000 years, maybe. So that pretty much says that no, we're not going to see a supernova from Betelgeuse. But now it turns out that we have another candidate for a star that could be a supernova. And this star is called AG Carina. I think that's the right pronunciation, but I'm not sure. It's located in the constellation Carina, which is a constellation, unfortunately for us, it's only, well, it's pretty much a southern hemisphere constellation. If you live below 20 degrees north latitude, you can see it sometimes. But basically, if you want to see it, you need to go below the equator. But at any rate, this is a star in that constellation. It's thought to be about 50 times the diameter of the sun. It's about 20,000 light years away. And as such, even though it's large, even in South America, you can't see it without a telescope because it's just so far away. Now, remember, Betelgeuse is what astronomers call a red supergiant star. It's red because it has changed from fusing hydrogen to fusing helium. That's a sort of a tip-off that a star is doing that. This star, AG Carina, is not a red supergiant. Instead, it's what's called a luminous blue variable star. That's an extremely rare type of star. Astronomers currently know only about 50 such stars. And they're also extremely unstable. Astronomers don't understand really very well what's going on with the stars, but they do know that they occasionally undergo really violent eruptions in which they throw out a large amount of gas from the star into the space around the star. And in studying this AG Carina, they think that it had an incident like that about 10,000 years ago. If we go back and look at the picture here, you can see there's the star there, and then there's this cloud around the star, which is the gas that got thrown out. And by measuring the distance between the star itself and this gas here, they can figure out that it probably happened about 10,000 years ago. Now, nobody has ever seen a luminous blue variable star that had two rings of gas like that. They found others. Some of them have not yet gone through that process, and so there's no ring of gas around them. And some are like this star with one ring. Nobody's ever seen one with two rings. And astronomers take that to mean that this only happens once, that a star can go through a process and throw off a bunch of gas. But the next time that it does anything like that, rather than throw off a bunch of gas, it just blows up. Now, we've got a little short video here, which gives you an idea of where the star is located in the sky. It's totally composed of Hubble images, and so we're going to run it just to see. Okay, so there's the constellation Carrier. There's the Southern Cross. If you've ever been below the equator, I know you've seen that. And now they're sort of zooming in on the area where the star is located. And in a minute, you're going, I think that's it right there. Now, these are all different Hubble images stitched together. So by changing its focus, it's able to get more and more of this star until finally we get in and we start seeing something looking like that original shot. So that's kind of what it looks like and where it is. Oops, sorry. Okay. So based on the fact that we know that this kind of star is very, very unstable, and we know it's already gone through one of these eruptions and thrown off a big cloud of gas, then it seems that at some point in the future, it is going to explode as a supernova. Now, the good news for us is since it's 10,000 light years away, there's not any danger for the Earth in that. If in fact it had already happened and we were just waiting for the light to get to us, we wouldn't have to worry about are we going to get blasted with gamma rays or anything like that, because it's so far away that we couldn't get any real huge amount of energy from such a supernova. But hopefully, at least if you were in the southern hemisphere, you'd be able to see it. And when I say see it, I mean see it with your eye, not require a telescope. But of course, we don't know whether this is true or not, but it seems like it certainly is a real possibility. Okay, anyone have a question about this story? Okay, we got one more today. Oh, I'm sorry. This is the last. This is also a different Hubble image where it sort of focused in on the star itself. And so you see a bigger gap between the star and the gas cloud there. Okay. Now, our last story says that a new species of cyanobacteria has been discovered. Well, since we talked about cyanobacteria, that interested me. So I took a look at it. We'll talk a little bit about it. This was just announced. A group of biologists said that they had discovered this new species of cyanobacteria. Now, you remember that we talked about cyanobacteria while we're talking about algae. Cyanobacteria are not considered algae themselves, but they play a very important role in the evolutionary development of algae. Cyanobacteria, what the unique thing about them is they're single-cell organisms that do photosynthesis. And it's the only known organism of this type. And their relation to algae is we're pretty convinced that billions of years ago, algae were unable to do photosynthesis. Now algae are multi-celled as opposed to single-cell organisms like cyanobacteria. But we think that algae at some point incorporated cyanobacteria, basically that an early algae, so to speak, ate a cyanobacteria as it sort of enfolded in around it and absorbed it. And the cyanobacteria took up residence in the nucleus of the algae, one of the algae cells, and basically stayed there and continued its function of doing photosynthesis. So we think that's how algae gain the ability to do photosynthesis. So that pretty much says that cyanobacteria are what we might call the father or parent of photosynthesis, the original photosynthetic organism. And as such, they are extremely, extremely important in the lives of us today, because before they came along, essentially there was not any oxygen in the Earth's atmosphere. The atmosphere probably then consisted mainly of carbon dioxide, like the atmosphere of Mars, and with a little trace gases of various types. But oxygen, if you took high school chemistry, you know, oxygen is chemically a very, very active element. Oxygen will combine with just about anything. And as such, if you go someplace where there's no oxygen and you have to take a jar of oxygen along with you and you open the jar and the oxygen comes out, then what's going to happen is it's going to find some other element to combine with. And after a short while, there won't be any free oxygen anymore. It will all have combined with other elements and formed oxides. So how do we get and keep oxygen in our atmosphere? And the answer is through photosynthesis. If it weren't for plants and algae and cyanobacteria, wouldn't have any oxygen here. And as I said, the evidence is pretty solid that cyanobacteria started this process. So the fact that we're here today having this class, we should all thank a cyanobacteria, because if it hadn't been for them, it would have never happened. So what's the story on this new species that's been found? There are a group of biologists who were studying a species of plant called a hornwort, which grows in Panama. And when they were doing their research on hornworts, they came across some cyanobacteria. And it didn't look like any cyanobacteria that they had ever seen. So they were curious. They said, well, let's check this thing out. And so they did DNA analysis on this cyanobacteria they found. And sure enough, it's different from any known species of cyanobacteria. It's most closely related to a species of cyanobacteria called Globobacteria. And that is the oldest known species of cyanobacteria. But when they look at, we got a picture here. This is an electron microscope image of this new cyanobacteria called Anthrocerotibacter, I guess. Again, apologies, I'm probably wrong on the pronunciation. Notice the size indications. That's 1 micron, a millionth of a meter. So the length is maybe 5 or 6 of those. And there's a cross section here. It's 500 nanometers. That's 10 to the minus 9 meters. So that's really, really tiny. So the other thing that they deduced from looking at the DNA structure of this new type of cyanobacteria is that it's related to this known type, but it seems to be more primitive. And so they're really thinking that they have come across what may be the ancestral cyanobacteria, the one that really got everything started. And so they're hoping that if they study the way photosynthesis works in this new type of cyanobacteria, the one thing that they've seen already is that it performs photosynthesis much more slowly than any existing cyanobacteria do, which sort of hints at it maybe, it's not that good at it. compared to other species of cyanobacteria. And so it might possibly be the first species to ever do photosynthesis. If that's the case, then by studying that photosynthesis process, they may be able to get an idea as to how photosynthesis started in the first place. Because that's one of those real mysteries for biologists. And it's, of course, a very important mystery because if it hadn't happened, once again, we wouldn't be here having our class today. Okay, anybody have a question about this one? Okay, well, that's our story for today. So I'd like to thank you all for joining us today, and we'll invite you back next week. I kind of think, I don't know, I've been following as best I can what's happening with the Mars mission, and it doesn't seem like anything big is going to occur over the next week that I can see. So next week may prove to be the first week in a long time that we really don't have a story about Mars. But come back next week and find out for yourself. So thanks everyone for coming, and I hope to see you then.
Thanks, Bill.
Thank you.
Yeah. Bye.
Thanks, Bill. Bye-bye.